Welcome to ThinkTech on OC16, Hawaii's weekly newscast on things that matter to tech and to Hawaii. I'm Jay Fidell.
And I'm Chris Lethem.
In our show this time, we'll talk with a world-famous scientist, Dr. Angel Yanagihara, a biochemical researcher at the John A. Burns School of Medicine at UH.
We'll hear about her training, her scientific research, and the discoveries she has made in her study of the Hawaiian box jellyfish over the past 18 years.
To learn about her science, we interviewed her here in the ThinkTech studios. We followed her as she collected jellyfish in the middle of the night in Waikiki, and then we took a tour of her high-tech laboratory at UH Manoa.
We all know that box jellyfish are common in the waters off Oahu, coming in after the full moon along the shores of Waikiki, Hanauma Bay, and other places on the island.
These jellyfish pose a serious threat to swimmers. They have long tentacles filled with powerful toxic venom that can cause serious pain and injury or even death.
The public should know about the dangers of being stung by these box jellyfish and how important it is to stay out of the water when they are spawning near the shore.
Dr. Yanagihara first came to our studio to appear in our Research in Manoa show, which broadcasts on ThinkTechHawaii.com every Monday. We asked her about how she got started on her work in this field. How in the world did you get to be a jellyfish scientist?
I went for a morning swim at 6 a.m. There were these little clear blobs on the beach, and an older lady was walking the beach, and she pointed them out, and she said, "You should not swim today. These are very dangerous. They're box jellyfish." I'd never heard of box jellyfish. I ran into a whole swarm of these box jellies. I didn't see them. All I felt suddenly were just burning needles injecting into my throat, and it was absolutely horrific. So my lungs were filled with liquid, and I was gasping and wheezing. I was in excruciating pain, and I just kept counting strokes to get to the shore. I ended up going to the shore doing everything wrong. I stood under a fresh cold water shower, that chain shower on the beach there, and everything just went off.
Wow, what an experience. Oh my God. Thank you for being so graphic about it.
Oh, okay.
So what did you have to do to, relieve yourself and get healthy?
This should be easy to manage. It couldn't be worse than a mosquito or a bee sting, etc. So I tried all the typical remedies and nothing worked. And that's when I did what they call a literature search to look to see what's been published and is there anything out there that's known, has it been acted on? And basically I found that, modern biochemical methods had not been brought to bear on this problem. I wrote a grant proposal to Hawaii Community Foundation, and that was funded. Hawaii Community Foundation grant involved getting an aqueous venom from the tentacles to look for ion channel blockers. Well, first of all, I found the venom, what people were calling venom in the literature, was really not very compelling. So they're taking freeze-dried tentacles, and you're going to put that into a blender with dilute saline and call that venom. So my approach was to take live animals, freshly caught from the ocean, and to develop a method to isolate these stinging organelles called nematocysts. What we devised is a method by which we can take the live tentacles and at 4 degrees refrigerator temperature, slowly rotate these in a specific solution which allows the tentacle to just retract a little bit, and these nidades fall out intact. The venom is still within the little capsules. So we can take those stinging cell capsules now that we've purified and centrifuge them, pellet them, and then I developed a method to rupture them in mass. And we hard spin out the particulate matter. Now we have the actual content of the nidade.
Okay.
And it was not at all what I had hoped. Basically, it's over 30% lipid. So if you imagine going to the McDonald's, getting a Big Mac, adding a cup of mayonnaise, putting that in your blender under liquefy, that's what you're giving to your gentle biochemist. Your biggest challenge is to be given an unknown goo, and your professor tells you, go solve that, right? So in fact, this venom was exactly that kind of challenge. But now, 18 years later, we can say that we've been able to really interrogate this complex cocktail, and we have done what's called a bioactivity-directed purification, which means that you separate a complex mixture using one method and then another and another, et cetera, and you get fractions. And each of these fractions you put down through this hot or not assay. So we were able to isolate a particular fraction that in and of itself could recapitulate death in a mouse or of human red blood cell lysis. The original objective was to be able to determine if there was a single bad actor. There may not be. There may be a number of them that work together, and then you have to knock out each of them. In this case, fortunately, it turned out that this one protein is necessary and sufficient for death. The fastest acting agent was this pore-forming protein. Then in your keyhole led me into another room and another whole dimension. We looked at how these proteins could interact, and then from that I had another keyhole moment, if you will, to look for ways to inhibit. My perspective is everything I'm doing is geared towards understanding diseases. So we call that pathophysiology. So how is it that a 1 meter sting of a box jellyfish can kill a child in 5 minutes? How does that happen? And what I found that goes into this pathophysiology of life-threatening stings is very similar to bacterial sepsis. So when one studies it from that end, then the collateral benefit is that things that could be addressing the box jellyfish sting could potentially also address bacterial sepsis. So rather than having the metric of let's discover things to cure cancer and to do this and that and to repurpose for our own good, if one sort of goes at it in a different point of view to learn from the data and to follow this thread wherever it will go, and to have a great tool set in which to do that, then the collateral benefit is that we actually start to understand bigger picture phenomenon.
She talked about her research into the box jellyfish and the innovative tools, methods, and techniques she's developed over the years in her pursuit of that research.
She told us about her collection of the box jellyfish, her data gathering and analysis of their tentacles and venom, her publications in the field, and her multidisciplinary approach to the development of this area of science.
Later, we met her on Waikiki Beach in the wee hours of the morning to watch her in her regular collection of box jellyfish, and she explained how she handles them. Here's a shot of the jellyfish she has collected in the past month.
We're out here this morning collecting box jellyfish. They come in each month, 8 to 10 days after the full moon. This is a particularly big, healthy specimen. We come out here to collect them so that we can cut off the tentacles and purify venom from these tentacles. Tonight is one of the biggest that we've ever had. It seems to be over 1,000 already, and because they're still coming in, I think the number might even get to 2,000. So we've just had a couple over the last 18 years that have been over the 2,000 mark. This particular species is nicknamed the Hawaiian box jellyfish, and it comes in every month along the lee side here, Waikiki, and often also Hanauma Bay and all the way up to Pokai Bay occasionally. But every single month, like clockwork, Landy Blair was the first one to really characterize this, an ocean safety lifeguard back in the 80s and early 90s. And when I was stung, he showed me the data. And we've been taking numbers and counts. We've been out here every single month for the last 18 years to do these counts and collections. So we now have the longest continuous data set in the world. Our work as biochemists has brought us to have to go out and catch the animals as they come in. So we've learned a lot about their biology. So we're interested to know why there are some big months like this, but so far folks don't even know the actual ages of animals, how long between spawning, which is what they're doing here, and they go off deep offshore, and then they come back once in their lifetime to spawn, and that's the end of their life cycle. So we're fascinated by that life cycle and trying to determine exactly what the age range is of the animals that come back. So they each have a organ, basically like a grain of sand, which grows day by day, and we can do some microdissection and then analyze that and determine how old they are. We basically use jewelers' techniques to grind that fine, and it shows like the tree rings, like rings of a tree, and we can count those under the microscope and determine the age of each animal. And from that we can determine, because all the animals that are coming in we've found by microscopy to be reproductively active. So they're all males and females that are adult, and they're spawning. So they basically have the sperms and egg, and fertilization is occurring and embryos are being dropped. This animal, for instance, is a very large one, and they range in size from just about half that size to this animal. So there's heterogeneity in the ages of the animals and also in the size, but it's a one-time trip. They only come to spawn once, and we've never seen them swim back out again. So they get pretty well beat up over the reef, and when we collect them, it's kind of rare to have such nice long tentacles. We're now looking to develop IV therapeutics for life-threatening stings, and I just got back from 3 weeks in the Philippines and 3 weeks in the Gulf of Thailand, where there are a number of deaths per year from box jellies. They have other species. There's over 30 different species of box jellies, and fortunately few, if any, deaths, well, at least 2 deaths have been attributed to this exact species. But there are other species of box jellies that are really racking up the deaths, and there are no effective treatments apart from the technology we've developed. The number one tips to avoid being stung is not to swim if there are jellies on the beach, or if the lifeguards are telling you that there are box jellies. You can see that they're absolutely transparent. You can't see these in the water without special lighting. That's what we have tonight. But the tentacles are vibrantly pink. It's hard to see as they're stretched out like this. But basically, you're not going to see what stung you until after the fact.
To follow up, we visited her laboratory at UH Manoa, and she gave us a tour and a first-hand look at what she has there and the research she does there. We stayed away from the tentacles and the venom.
We have basically everything in this laboratory to go from keeping a live marine animal for a few days, to purifying the venom, to doing the bioassays, to preparing potential inhibitors and treatments that can be tested for the ability to treat stings. This week we were doing some experiments with live animal tentacles, and we tested some of the common things that are out there, the effect of shaving cream, hot packs and cold packs, as well as our own technology again. Here are tentacles in seawater, and they're still alive. You see how they're pink? If you put them on your arm, you would be stung instantly. Yeah. This is blood agarose, and these clear zones are where the venom has been injected and is lysing the red blood cells, and that's why it's clear. If we take another piece of tentacle, for instance, this one, and we lay it back on the blood agarose again, you can see right now it's contacting. If we go look under the microscope, you'll see that the nidae are discharging. This is made with human live blood and the blood agarose. So we leave that on for 5 minutes at 37 degrees. It's warm, like your body would be. And after 5 minutes, we remove the tentacle and we do different treatment. So we see if our treatment reduced hemolysis or increased hemolysis. And some of our treatments massively reduce hemolysis, and some things make it make no difference or even make it worse. So this is a method that we invented. So it's a novel contribution on our part to the field. Part of the problem in this field is that there aren't enough studies, there aren't enough labs doing it, and it has historically required human volunteers. So there's where the tentacle was. So you see that streak there? This other one to the right was on there, was in the incubator for 5 hours. So I'll show you what the nidae look like. Where I laid the tentacle, there's a track of the tiny stinging capsules. They're left stuck on the surface. Each of these tiny nidae are filled with the venom. So whenever a tentacle contacts your skin, many undischarged nidae are left there. So the red blood cells are having holes pierced in them, hemoglobin seeping out, and so there's a clear zone. So you can see both where the nidae were left on the agarose, just physically left there, as well as the clear zone of the activity. This assay looks at one particular one, which we find to be the worst offender and to be necessary and sufficient for causing death in a mouse or in a piglet and presumably in the human being. So that's why our technologies have all been dedicated towards inhibiting this particular activity. Most of the deaths due to box jellyfish occur in the Indo-Pacific, Northern Australia, Malaysia, 20 to 40 a year in the Philippines and Gulf of Thailand. So recently I spent 3 weeks in the Gulf of Thailand on one small island where 6 deaths have occurred just over the last 2.5 years. And I also spent 3 weeks in the Philippines where we have supposedly 20 to 40 deaths per year. So we're interested in working in places where there's a lot of loss of life to provide our technologies and to do these kinds of bioassays we just showed you today with the local prevalent species. So we want to take our model of human tissue, which we just showed you, to Thailand and Philippines, and use the local animals there that are causing death in children and tourists alike, and see, again, what are the best ways to inhibit the venom once it starts that destructive pathway.
In the course of discussion, she gave us some tips on how to deal with being stung by jellyfish and how to counteract the effects of their venom.
We recommend that you first saturate the site with the spray because that will inactivate any of the undischarged stinging cells, and then prepare the site for the cream. The pain scores go basically down to 0. From 10 to 0 to 2 in 7 minutes. This is one of the figures from our recent publication, and we compared different ways to rinse a sting site. And you can make things worse by choosing the wrong rinse. You'll remember in my story, the cold fresh water made things worse. Here we show definitely that that's happening in a model that we've designed in our laboratory. So we found that the best rinse as far as improving outcomes is the Sting No More spray, and next to that, a good rinse is vinegar. As far as treatment goes, we compared no treatment with seawater, freshwater, vinegar, isopropanol. Ethanol makes things worse as a treatment. Urine actually made things worse. Epsom salts made it way worse. Hot water improves outcomes. The current practical summary then, based on this 18 years of work, is that the best approach is to avoid them. If you're stung, research shows that there's potent inactivation of these stinging cells by the Sting No More spray, and then treatment and inhibition of the venom already injected into your skin by this pharmaceutical grade Sting No More cream, which was produced University of Hawaii at Hilo College of Pharmacy, chief pharmacist for a Department of Defense grant. If one doesn't have the Sting No More spray available, vinegar is a good rinse, but it isn't a treatment. Things that aren't good and should not be used are cold packs, and do not do what I did, which was stand under the fresh cold water. Interestingly, hot fresh water is not a problem. It's the cold fresh water. Do not scrape the site. EpiPens can be dangerous and life-threatening.
Dr. Yanagihara's research goes beyond the Hawaiian box jellyfish. It also examines other dangerous box jellyfish in other places in the Pacific and Asia. And her techniques and discoveries are of value all around the world.
Kudos to her for her remarkable work and world-class research in this field of science, and of course for her development of the unique Sting No More anti-venom product.
We all need to follow what is happening in science at UH Manoa. We need to recognize, encourage, and support Dr. Yanagihara's work and the work of other dedicated researchers at UH who strive to make it a center of excellence in today's global scientific community.
And now let's take a look at our ThinkTech calendar of events going forward.
ThinkTech broadcasts the video and audio of its talk shows live on the internet from noon to 5 p.m. on weekday afternoons. And then we broadcast our earlier shows all night long. And some people listen to them all night long. If you missed a show or you want to replay or share any show, they're all archived on demand on thinktechhawaii.com and on YouTube. If you want to listen to our audio stream, go to thinktechhawaii.com/radio.
Visit thinktechhawaii.com for our weekly calendar and our live stream and YouTube links, or better yet, sign on to our email list and get the daily docket of our upcoming shows.
ThinkTech has a high-tech video studio at Pioneer Plaza. We invite you to come down, see our studio, and be part of our live audience. Contact me, Jay, at thinktechhawaii.com. Be a part of our civic engagement here on ThinkTech.
Go ahead, give us a thumbs up on YouTube or send us a tweet at ThinkTechHI. We want to know what you're thinking and how you feel about current issues and events affecting Hawaii. We want you to stay in touch with us, and we want to stay in touch with you. Let's think together.
On March 17th, ThinkTech, in conjunction with the Grassroot Institute, will present a Downtown Forum luncheon panel at the 6th floor auditorium at 1132 Bishop Street. Our program is called Beyond Politics, Pathways for Hawaiian Advancement.
The program will discuss the issues facing the Native Hawaiian community and will explore alternatives by which that community can achieve its cultural, social, and economic goals other than through restoration of political sovereignty.
Our frame of reference is that quite apart from the question of political sovereignty, there are several pathways by which Native Hawaiians are advancing, such as education, entrepreneurship, political involvement, and so forth. We'll present a panel of speakers who can address those separate pathways.
Join us and raise your awareness about the critical changes affecting and taking place in Hawaii. Be a part of the conversation and sign up to attend on thinktechhawaii.com.
Want to speak out about a community issue or event? You can. ThinkTech invites you to come down to our studio and make a video at what we call our Speaker's Corner. If you'd like to express yourself, contact me, Jay, at thinktechhawaii.com.
We'll be right back to wrap up this week's edition of ThinkTech, but first we want to thank our underwriters.
Okay, Chris, that wraps up this week's edition of ThinkTech. Remember, you can watch ThinkTech on OC16 several times every week. Can't get enough of it, just like Chris does. For additional times, check out oc16.tv.
For lots more ThinkTech videos and for underwriting and sponsorship opportunities on ThinkTech on OC16, visit thinktechhawaii.com. Be a guest or a volunteer, a producer or an intern, and help us reach and have an impact on Hawaii.
Thanks so much for being part of our ThinkTech family and for supporting our open discussion of tech, energy diversification, and globalism in Hawaii. You can watch this show throughout the week and tune in next Sunday evening for our next important weekly episode. I'm Jay Fidell.
And I'm Chris Lethem. Aloha, everyone.